just wanted to review some of the immune cells that we have talked about and their role in immunity and potentially in disease, but mostly in protection. We will get more into autoimmunity and how specific cell types can cause specific diseases. For right now, I want you to just focus on general life cycle of these cells, and by life cycle I mean just general purpose, what they're trying to do. So on this side I drew the granulocytes. Here I have the phagocytic cells. This is an overlap right here. Neutrophils are both granulocytes as well as phagocytic cells, and then the lymphocytes being the T cells and the B cells. Remember, all cells that you see here are called leukocytes. Okay, is an E. Okay, so granulocytes. And that is, again, granulocyte is referring to the granules produced in the cells that have toxic effects against pathogens. And then these are the phagocytes, meaning that they will engulf pathogens. They can engulf entire pathogens, or they can, if there's cell debris of any kind, either from the pathogen or from our own cells, such as if a virus lyses one of our own cells, like what happens when you get a cold. Cell lysis by the virus will expose internal proteins that can be eaten up by phagocytes and then presented to the immune cells, which then, presented to the lymphocytes, which can then activate the immune system. So there's multiple pathways and multiple ways, all of which play a role in activation of the immune system. So over here, actually all of these, of all the leukocytes excluding T cells and B cells, are going to be non-specific, and they will search and destroy, and that is essentially their only purpose. For the phagocytes that are in green, they play a larger role in activating the adaptive immune system, so the T cells and B cells, than do these other ones. However, they all do play a role. So, for example, if we have a pathogen, there's a couple different things that can happen. So if I have just a neutrophil or just a macrophage or just a dendritic cell that is capable of engulfing this cell. So this is, I'm not going to draw the whole cell. Remember, our cells are much bigger than bacteria, so this is the beginning of phagocytosis is occurring right here. So let's just say that this is a macrophage. The next thing that's going to happen is once this full bacterium is engulfed, it will be degraded by the reactive species that are found within the macrophage. This occurs also within dendritic cells and neutrophils. I just chose macrophage for no particular reason. And so once inside, so we're going to zoom out a little bit. So once inside the cell, you're going to have it's called a phagosome, because it is it occurred just after phagocytosis. As this is getting engulfed, there are other lysosomal, like granule-containing vacuoles that will be added to this phagosome, and it changes names to the phagolysosome. That is just to help keep track of basically what's happening. But what these contain are, like some examples, they can have lysozyme, which is targeted to degrade bacterial cell walls. It can have reactive oxygen species. So this will include things like hydrogen peroxide, chemicals that resemble bleach are found in them. they will play a role in destroying like the DNA and essential bonds that help keep the cell wall together, and this will basically just help dissolve the bacterium. Once the bacterium has been dissolved, then the pathogen will have its bits spread around. I'm just going to draw some bacterial pieces here. These pieces will be presented on MHC molecules, which we have gone over, and at the very least you should be familiar with what MHC does. If you still are having a hard time remembering 1 and 2, class 1 and class 2 MHC, that is okay. That will just come with time and repetition and increased exposure to the topic. Okay, so because this was a phagocytic cell, the pathogen is considered extracellular, and it will then be presented on MHC class 2. So MHC class 1 is for intracellular antigen presentation. If it was engulfed like this bacterium was, it will always be presented on MHC class 2. It's then that this MHC presentation is presented to, or is taken to T cells, which then are able to recognize basically whether or not the things that are being presented, the antigens that are being presented, are pathogenic or not. So here I have a T cell being presented antigen by 2 macrophages. One of them is presenting bacterial antigens. So from the previous page, we engulfed our bacterium, chopped it up, presented it on MHC class 2, and now we found a T cell that recognizes, that has a similar binding site to the bacterial antigen. At the same time, there is a different macrophage that found some debris that it phagocytosed that were actually human cells. It was part of our own cells, so it was collateral during the bacterial infection. Some of our own cells died, and their debris was taken up and eaten by a macrophage and is being presented also on MHC class 2 to a T cell. Well, fortunately, T cells are trained to recognize, again, this happens during maturation. T cells are trained to recognize basically molecular patterns that are found only in pathogenic species, bacterial or viral or fungal. They're trained to recognize those patterns from molecular patterns from our own cells. So there are like certain amino acid sequences that are only found in human cells that T cells are able to recognize and say, no, those are ours. So this actually will not initiate an immune response. It will, but not an adaptive immune response. And when I say it will initiate an immune response, it's not normal to have our own cells phagocytosed and presented. This is always occurring at a low rate because there is always cell death of healthy cells, and that's good. But the T cell will basically say, no, don't continue attacking this particular antigen. These are okay. Keep doing your job. Look for something else. That being said, of course, there is a caveat. In order to this will promote certain types of immune responses that will help regenerate dying cells, and macrophages do play a role in helping clean up dead cells because dead cells are great food for bacteria. So this is still an important process, even though this X represents stopping the adaptive immune response that will not occur if self-antigen is presented. Bacterial antigen, on the other hand, or viral or fungal, if presented to the T cell, the T cell is able to recognize it and say, hey, these are not our cells, and something is here that's not supposed to be. So it will activate this macrophage to continue doing what it's doing, but better. It will do a better job at phagocytosing to basically eat up any bacterial cells that it sees and presenting them to trigger additional T cell responses. And remember, T cells are required for the initiation of an immune response. No adaptive immune response can occur. Okay, of course, there's a caveat. Most adaptive immune response cannot occur without T cell activation. So this interaction has to occur. And so these are the T cell receptors that are capable of recognizing and binding to antigen, same as the MHC class 2. What also has to occur, though, is that this protein, this surface protein, this is the CD4 protein. So remember, 2 and 4 go together, and then cytotoxic T cells are, I'll just write it up here just as reference, so those are CD8 cells. It looks, for our purposes, identical to this, except for this protein is going to be called CD8 instead of CD4. So CD8 cells recognize MHC class 1. And it's a similar activation system. It just depends on where the antigen being presented originates. Intracellularly, MHC class 1, or extracellularly, MHC class 2. So antigen recognition by a T cell is what activates the T cell. Until this interaction occurs by an antigen presenting cell, so it could be a dendritic cell, a macrophage, or B cell, once an antigen is recognized and activation occurs through either CD4 or CD8, depending on the type of MHC, naive T cells, as they are called, become active T cells, which then start differentiating, so replicating into effector and memory cells. Memory cells remain dormant until the next exposure by the same, the next exposure to the same antigen. Effector cells are going to start differentiating into T helper cells, so I'll just abbreviate it like that. T helper cells. There are two kinds of T helper cells. Actually, there's multiple kinds, but two main kinds, TH1 and TH2. Don't worry about differentiating between the two. Just know that they arise after activation of a naive T cell. And these are going to activate additional immune cells. So T helper cells will then do things like activate innate immune cells. So things like, again, like macrophages and mast cells and monocytes, eosinophils and basophils. All of those things will become activated by signals secreted by helper T cells to come to the area of the infection and basically to do their job. So all adaptive immune response, for the most part, will have to occur through T cell activation. There is a T cell independent reaction that can occur. So even B cells that recognize antigen and then present them to T cells require activation through this type of interaction. B cells are also going to present antigen to T cells on MHC class 2. However, if there is significant binding to, in this case, LPS, then B cells do not require T cell activation. So this is called the T cell independent response. So remember, endotoxin is a really powerful inducer of the immune system, and this is part of the reason why, because they're long lipopolysaccharide chains that have multiple binding sites on them. If you're unfamiliar what LPS looks like, it is in a couple of my slides. The one from this past Thursday, I think it was like the 18th or 19th. The 19th. So, anyways, they're long chains that can bind multiple sites. Because they're repeating subunits, that's why they're capable of binding multiple epitope binding sites on B cells. If this occurs, remember, as bacteria proliferate, they shed this LPS into your serum, and this can bind and activate B cells because so many of the antibodies are being activated at once. Activated meaning they are binding their antigen. So when this occurs, B cells can actually forego T cell antigen presentation and start proliferating into, same as T cells, into effector cells and memory cells. So memory cells, again, are going to remain dormant until the next time the antigen is present in your system. Effector cells, an example of this, are going to be plasma cells. And these are just antibody producing B cells. And these are just going to pump out tons and tons of antibody that are going to play an important role in opsonization of the pathogen for recognition by non-specific immune cells as well as additional B cells and T cells. And so I've used the word multiple times, but this is all part of a positive feedback loop, so that the more activation occurs, it causes more immune cells to get involved into the system. So it can get out of hand really fast, but luckily there are regulatory T cells. So regulatory T cells, that is a T. Oh, I just keep making marks all over the place. Regulatory T cells. That will secrete cytokines that deactivate all of the activation that has occurred. And so these are really important in stopping infections from, or not stopping infections, but stopping the immune cells from causing too much damage because they're so powerful. Because remember, as they are secreting antibodies and cytokines and granules and all these things meant to attack pathogens, there is collateral damage from our own cells. Many of our own cells do die, and a lot of the symptoms you get from sicknesses are actually not from the pathogen itself, but from the immune response doing its job too well, essentially. So regulatory T cells are going to use cytokines. Cytokines. Just to see that word. It's very bad. These are going to secrete cytokines that will basically tell everything to calm down. And